Hi, I'm Mike Okimoto from Labcompare, and I'm talking with Stuart Shand from Olympus, who's going to talk to us a little bit about the VivaView FL Incubator Microscope. Hi, Stuart.
Hey, how you doing?
So, the first thing that comes to my mind is this doesn't look anything like a microscope, in fact.
Right. So what we've actually done is we've taken all of the components that we use in our regular microscopes and actually incorporated them into an incubator. When cell biology departments or researchers are working with cells, they grow them in the incubator. They then transfer them to the microscope. What we've done here is incorporated the components into the incubator, so there's no need to have to remove them.
Okay, so essentially the guts of the microscope are inside this box here. Can we take a look at that?
Absolutely, yep. So the idea is that we have no compromises in the level of incubation that we're able to offer to the cells, particularly important for sensitive cell lines like primary neurons, but also for long-term time-lapse imaging where it's really important to have the utmost in incubation, and the way to do that is with an incubator.
Right, so in that case you don't have to take your culture in and out of the microscope, you're not constantly changing temperature.
You're not going from the 37 degrees down to room temperature and then back up and putting the cells through these temperature cycles. They stay in here and are imaged in here.
Excellent. So how does this work? So
the stage that we have here takes up to 8 samples, so we can do multiple conditions, controls, different treatment groups all at the same time. Each position we can set up multiple positions within that dish, so we're not just limited to 8 fields of view, we can do multiple within each of those, so it's really increasing the efficiency in the number of cells that we can be monitoring.
So you're taking a bunch of different data points essentially from one culture.
Absolutely.
Excellent.
Yep.
Okay.
So it's an inverted microscope, so the objective is in there beneath the stage. And what we've done is we've actually enclosed the objective partly inside the incubator as well. It's sealed away from the humidity, but it is heated. So everything inside the incubator is at 37 degrees. The objective is at 37 degrees, so it means there's no temperature gradient.
Okay.
So
no condensation worries.
No condensation, but also the important thing there is no focus drift. So as the temperature of the room is changing, which would historically, or with some systems, it causes fluctuations in the focus of the cells. With this, we're able to maintain it over the long term.
Okay, and what type of objective does it come with?
So it comes with a 40x U-Plan S Apo. It's a high NA objective, 0.95.
Okay.
So the idea again is we haven't compromised incubation. We certainly don't want to compromise the level of imaging that our customers are familiar with all our other systems. So it's a really nice objective in there as well.
Okay. Well, what about the dishes themselves, and how you're taking the images up from the cells in the dishes? So
it's designed to work with 35mm dishes, and the important thing is the glass bottom. Again, to do high quality imaging, it's important to have that glass, that cover slip in there. It's like the final optical element in the system.
Sure.
Being that it's glass, we're able to do DIC, whereas other systems have been, they have to use phase contrast. So DIC means that we can do really nice high resolution contrast imaging. And then we also have fluorescence. The system comes ready, when it's installed, comes ready to do GFP and RFP.
Okay.
But we can go up to 5 fluorescent channels.
Oh, wow.
And any filters that a researcher may have in their lab, they can just be incorporated into a regular Olympus filter cube.
So if it works on your standard Olympus microscope, it'll work in this.
Exactly. And then any of the filter companies, if they have, as dyes are evolving and they come out with these new filter sets, then we just need to get it into one of our cubes and we can start imaging with it.
Interesting. Okay. Now I know one of the things that you said that this is good for is drug studies. So you can add compounds to your cells and then image them over time. So how can you do that? So
through the software, we have the ability to actually take the stage position that we're interested in, that we want to add the drugs to, and transition it around to the front of the microscope. So I can just go in and click the little software panel here, and what it would do is it would move the stage around to the position that you selected, and then what it does is it moves and actually opens up the dish.
Oh, look at that,
so we can come in here and you can actually manually inject and add drugs and there's no loss from the environment.
Right.
So with, conventionally we've had to open these systems up, then we're losing the environment, and that's what we're trying to maintain the whole time. We really don't want to disrupt the CO2, the humidity, and the temperature to ensure that our cells, the cells stay healthy.
So now you mentioned the software, so I assume that the software package that comes with it to analyze the images, or to capture and analyze the images, or what is the software package?
So we've actually worked with Metamorph.
Okay.
So we have Metamorph for Olympus, and they've specifically designed a user interface for us to control this system. With it being a circular stage, it was important to make sure that that was easy to use,
right.
For setting up the multiple stage positions. So very easy to use. Anyone who's familiar with Metamorph is going to be straight on it and using it immediately.
Right.
For anyone who's not, it's got a very intuitive way of just going through and setting up the experiment, adding the different positions,
right.
setting the exposure times. And then of course with Metamorph, it's got the very powerful post-acquisition analysis capabilities.
All right. Well, this is a pretty slick system. I guess the only other question that I have is, if I'm interested in getting this in my lab, what are the setup costs, or what are how much time is going to be involved in setting something like this up?
Yeah, well, it's really easy. It comes with everything. Everything's included. I didn't mention earlier, but the camera. We have a high quality CCD Orca R2 camera from Hamamatsu incorporated inside, and that's included with it. The light source is the Expo Exact. It's their new high-end system that's specifically tailored towards live cell imaging. And it's important not just with incubation. If we're going to have these cells living for the long term, we want to minimize any phototoxic damage that could be happening as well. So that's, we can control that in 1% increments, so we can get down to really low light level imaging, so we're not harming the cells. The camera just connects on as a regular camera would. The light guide and the adapter there just connect in nicely, and then everything is controlled through the software. So very easy to set up, and then to answer your question, the setup costs: cells on dishes, and then a CO2 supply, and you're ready for some long-term time-lapse imaging.
All right. Well, excellent, Stuart. Thank you so much for talking with us. For more information on the VivaView FL Incubator Microscope, click on the links below.